Okay, everyone. So my name is Zach Zhen, and today is a great honor for me to introduce Professor Arieh Warshel, a distinguished scientist in computational biochemistry and a pioneer in developing methods to study biological processes in the molecular level. Professor Warshel was awarded Nobel Prize in Chemistry in 2013 for his groundbreaking work in the development of multi-scale models for the complex chemical systems. He has published over 500 papers and received numerous awards and honors. He is a member of National Academy of Science and the Royal Society of Chemistry. Now, let's welcome Professor Arieh Warshel for his talk today.
Okay, it is a pleasure to be here and to be again in the molecular frontiers. It will be very hard to compete with the previous presentation. I could sing you in Hebrew some songs on rain and water, but you don't know them, so you will. If I knew this story, I would have brought some music. So this won't happen. And my connection with water started essentially quite early. In 1965, as an undergraduate project, I worked in the early reverse osmosis project at the Technion as a summer student. If this technique would have been adopted in California, we will have much less problems, but this is it. I would not tell you on what they spend the money. So what I will try to do is to describe modeling of water and related electrostatic effects in biology. And the perspective will be quite different than the previous speakers. Essentially, in many, many cases, we had to simplify the water, and I will justify it because, as we saw in the last lecture, when you try to study electrostatics, at the end of the day, different water models give you very similar effects if you know what you are doing. So I will describe modeling of water in treating large molecules. And it started around 1975 when we started to model enzymes, and we found that nothing happens, no bond is breaking, let alone no catalysis, when you don't have electrostatic effects which include water. So we had to model water and to model a lot of water, all the water around the enzyme. I had no interest, just a minute, let's see if the time is here. I had no interest in using continuum model because I could not put my reputation on something that I don't understand and don't believe, though I read all the books. So I came up with a very simple model called the Langevin dipole model. And the idea was as follows. If I want to capture the electrostatic effect of water, I could assume, or if I knew the field on each water molecule, if somebody told me, I could have modeled the water by dipole and parameterized the polarization of the dipole toward the charge by looking on observed solvation experiments. We've done this, and This model was extremely effective. I first presented in a Faraday discussion. Stillinger was the moderator, and nobody understood what I'm talking about. But we start to use this model, and it evolved to what we call protein dipole Langevin dipole, which says as follows. Remember, this is 1975. We have a grid of points. On each point, we put a dipole. The dipole is trained to have the same polarization as water at this point, and we calculate the electrostatic interaction between charges in the protein and other groups to these dipoles and between the dipoles to themselves. Needless to say that this model and subsequent models which represent the dipoles like sphere will receive unbelievable negative criticism, starting with everybody knows that water is not a dipole, which of course I knew, but what they miss is that you could train a dipole to have the same properties as water in terms of electrostatic effects, and in later things you could even have spherical dipoles which would create the equivalent of hydrogen bonding. Anyhow, this model was used in this QMMM 1976 paper to calculate the catalysis in lysozyme. And essentially, it's being used by us even today. But in the meantime, we had to respond to criticism and to computers become stronger. And remember, our main constraint was we wanted to get results with the available computer time, which were, of course, extremely weak at that time. So the next model was a spherical model with explicit water, which were also parameterized on crystal of water. And the main feature of this model was that it include a really small number of water in a sphere, so we did not have to deal with the problem of doing things periodically, but it include the entire universe. The surface of the sphere was trained to have the polarization of water at this distance, and then it was surrounded by bulk. And I remember that in one of the first rejection of this paper, which was rejected 3 times, the referee wrote, "You don't have a complete Hamiltonian." Until today, I have no clue what he meant, but obviously I have no clue that it's important to represent the entire system and the entire universe. So with time, we could consider several models. One is huge number of explicit water and the biological molecule inside. Now people try to use it. It's very expensive. The other is this grid of Langevin dipoles. And the other one is a model which is really a continuum model where you represent the protein as having dielectric 2, or whatever dielectric, usually incorrect one, and bulk around it. So with these models, since the, let's say, 1978, we started to explore properties of different electrostatic elements in proteins. This is the new drinking device we got here. Very, very inconvenient. Okay, so for example, if we want to calculate the energetics of a charge in a protein, you have to do a thermodynamic cycle where you, of course, surround the charge system by water. Then you try to calculate the energy of uncharging, or let's say if you have an acid, of putting proton on the acid. And then you complete the cycle. You could complete it moving the charge to water. This will be when you calculate binding energy. And you complete all of this cycle and get the energy, which is a very large energy. When you have aspartic acid, it's around 70 kilocalories per mole. And also here we had problems because most of the literature come from studying very small effects, like temperature effect on charges. But when you try to calculate the solvation of a charge, you are dealing with 70 kilocalories per mole, and the details do not matter so much. 1 kilocalorie per mole mistake is a fantastic model for this type of questions. So these are typical benchmarks when you try to calculate the pKa, which is basically the energy of taking a proton from an acid, in different sites in small protein, in this case BPTI. You do it by free energy perturbation, which means that you slowly change the charge from -1 to 0, and you watch how the free energy is accumulated, and you get a number. If your model is correct, and it involves a lot of factors, it involves in polarizability-induced dipoles, it involves charged dipole, and it involves the interaction with the water. Here I present the contribution for different sites, and you see that in different sites you could get very different contributions. But the nice things with water and biological interior, that there is a huge compensation. This is part of what we call dielectric, and at the end of the day, you get small energy. But it is very important to be able to capture the compensation and to be able to get the final number. Since when we use full microscopic dipoles, we had sometimes large mistakes. And since the competitions that use continuum got fantastic result, or so they claim, because if you calculate pKa, what people did not realize, if you just calculate it with continuum, and if the group is close to the surface of the protein, you will always get pKa which is very similar to the pKa in water. And you will say, oh, I have a very small error of ½ kilocalorie per mole, when this guy Warshel gets 3 kcal per mole mistake. So eventually we become tired and also add a dielectric to our proteins, but it's a dielectric that we could explain what it is. We could show how it represents the rearrangement of the proteins and other factors. So we have PDLD, which is pure microscopic dipoles, and PDLDS with subscript LRA, which means that we also calculate the relaxation of the protein. And this model appeared to be extremely effective, especially when you look on what matters. You don't look on group on the surface of the protein, which was the favorite benchmark of all people, but you look mainly on groups with large pKa shift. And this is where we used to do extremely well. This model is still our favorite model for calculating binding energy. It works better than most models in drug design. Okay, but now we turn a little to other systems. One of the systems that we studied the most at that time was redox proteins. Redox protein is also an electrostatic problem. You change a charge, for example, in a heme. Though when I try to publish my papers on electron transfer, a very prominent Redox person screams that from his NMR he knows that it's not electrostatic problem. Again, this is not a repetition of my problems with referees, but here is cytochrome C. We were able to reproduce very nicely the redox in this system and to show that what control it is the fact that the interior of the protein is not very polar. Then we launch a major project on many redox proteins focusing on iron-sulfur proteins. And not only that we got very nice correlation, we got some insight on the role of water inside proteins. Because in two proteins, which essentially are shown here, which looks identical to the crystallographers, not really identical, but very similar, the redox was very different. And this was a puzzle that had to be solved probably by theoretical approach. And of course, we found that there is 1 or 1.5 water more in one of them, which led to this very large redox shift. So we used this model in the 70, 80, 90 for asking different questions on electrostatic in proteins. And then we got a very challenging problem, which was introduced by Bertrand Garcia Moreno, who took Staphylococcus nucleus and asked the question, what happened to charges in the interior of a protein, where if the protein is an oil drop at some region, okay, let's put it like this. If you take region of the proteins which are very hydrophobic and try to put charges there, you will have enormous instability. It's like trying to have a drop of oil and to put inside a charge, and the oil probably will explode. It's a nice experiment to do. So he mutated the interior of Staphylococcus nucleus, put acid in places where there were no acids, and there was significant, very significant shift in pKa, moving from 4 to around 8. And the question is, could you reproduce it? So first of all, the beautiful thing about it, that all the continuum models, which people always like to use something which appear in the literature, all the continuum model has one problem. They depend on a dielectric constant, and nobody knows what is the dielectric constant that should be used. I will comment about it shortly. So we tried to see if we could calculate the pKa shift, and what we found, and I will show you two cases, Staphylococcus nucleus and cytochrome C oxidase. So if you try to calculate the pKa by charging this group, which is set in non-polar environment, and we do it with polarizable and non-polarizable models, you cannot reproduce the pKa shift. You get pKa of 20 or more because the charge apparently is unstable, but I told you the pKa is 8. So first question is, how do you model this? To me, it was clear that either there is local unfolding or there is water penetration, and you have to be able to model it. So the first model we came with was called the overcharging model, and the idea was as follows. Instead of charging it to 1, which does not do anything to the protein, we will charge this group to -2. Now there are huge forces on the protein, and the protein will start to unfold, or water will start to zoom in. And then we will return it to -1. So with this approach, one coordinate is the charging, the other is the coordinate change of the protein. We were able to force the protein to respond to the charge, which involved a barrier that the force of -1 was not enough. When I say not enough, it's not enough in our calculation time. In reality, it is enough. And when we done it, we succeed to reproduce the pKa. The protein show opening and penetration of some water. I don't like to show table, but if you could read it, even polarizable model did not help. Only the row before the last one reproduced the experiment, and this is the one where we overcharge the protein and then move back to -1 and got the result. So this approach is very fancy, also very smart, but we try to get something simpler and to ask the question, could we ask the water to penetrate to site in the proteins where they are in reality, but it's difficult for them to do it in regular simulation time? So this is a very challenging problem theoretically, how you let the water penetrate, especially penetrate to the right place, and then calculate the correct energetics. And we don't like approach that require, 1 month's computer time. So we always like to have 1 day at most so we could look at the result. Now, in principle, what you should do is what is called grand canonical approach, which means that you have huge box in the bulk, and the water could try to go to the protein, and in principle, you do it by Monte Carlo insertion, and you try and try and try. Unfortunately, most of the attempts fail completely, because in most cases, the energy where you try to insert the water is higher than in the bulk. So in the first few years, eventually we also did grand canonical, and I will show you something, but we designed this water flooding approach, which was, again, a nice trick, and it goes like this. Instead of trying to insert the water all the time, which would take huge amount of effort, we insert very large number of water to the protein by brute force, let them relax, and use what is called linear response approximation, which is the first term in free energy perturbation. It's the term, where do you see it? So it's the term in the lower part of the slide. And from this, you could get the free energy of each water molecule in this over-flooded cluster of water. And you could also calculate water-water interaction. And then we run Monte Carlo, not on the insertion to the proteins, but we try to generate different cluster of water just by knowing the energy of each water and each pair. So we run it in a very small subroutine, what usually would have been the grand canonical calculation. We get a result, which I will show you is quite reliable. So we do it in some cycles that we don't want to consider. And we try it, for example, in this mutant of Staphylococcus nucleus. And as you see, without it, if you just put X-ray water, the pKa is very bad. When we use this approach, we get very close to experiment. Another case, where we go to cytochrome C oxidase that I will consider later again. There is a channel there, the D channels, where protons are supposed to be going through. There are groups that are supposed to accept the proton. There are in sites which are not so polar. So we succeed to insert water there, and again we got close to the observed pKa by this approach. Now this was around 2012. By the way, this approach is extremely important in drug design, and large part of the selling pitch of Schrödinger is that they could do it. First of all, their approach has conceptual problems. Second, this approach is much better, but we don't try to sell you anything. So now, around 2018, we decided that we have enough computer power to try to do grand canonical approach and to see how well this water flooding approach works relative to grand canonical. And this includes questions. Could we find waters that are found by X-ray in special sites in, let's say, BPTI? So first of all, what is the approach? What we do, we have bulk water, but instead of waiting for the case where the water will be happy enough in Monte Carlo to go in, we apply extra force that force the water to go in. So when we change this force to larger and larger value, we get more and more water in the protein. In this case, we have 3 water molecules in BPTI. And what we compare here is the result with this water flooding and grand canonical. We get similar results. Also, we get similar positions for the water molecule. Now, what is this? Okay, we still. So we have a way to insert water and to reproduce their position in the X-ray, and more importantly, to get the electrostatic energy, including the effect of internal water, which is extremely important, for example, in cytochrome C oxidase, where we try to find out what is the pathway of the water. A very short comment on the dielectric effect. We spend huge amount of effort on figuring out the molecular basis of the dielectric effect. We wrote very instructive reviews about it. Some of them are understood. So one of the most important effects of dielectric is when you separate 2 charges, let's say in water, you do very small amount of work, and the physicists like to describe it as screening, which is very confusing and not so useful description. The best description is compensation by solvation. If you separate 2 charges, when they are at very large distance, they are stabilized a lot by the solvation of each charge. When you bring them close together, they are stabilized by the Coulombic interaction. In nature, because there are a billion configurations of the water and also of the protein, there is this compensation. This is why, even if we don't know the exact structure of water, their main physics, in my opinion, is the compensation of polar material. It's this having many, many minimas, which give you small free energy for different changes. So this is dielectric for charge-charge. We also spend a lot of effort calculating the dielectric in protein using the definition of the dielectric constant, calculating the macroscopic field for microscopic calculation. And we established that the dielectric in protein is not 2 and not 4, but frequently very high, approaching 10. And again, I could have given you a very long discussion, but one thing that you have to realize that the interaction between two charges in proteins is very largely compensated by rearrangement of the protein dipoles and the solvent. And now we go to, okay, this is just comments on water in enzymatic reaction, where in addition to being a solvent and providing dielectric effects, sometimes the water acts in a very intimate interaction. This is, for example, subtilizing. When you mutate polar residues, a water molecule almost completely compensates for this effect. There are cases where the water is part of the reacting system. For example, this is the reaction of Ras P21. Okay, so this is a case where a water molecule attacks a phosphate, breaking GTP to GDP. The green one is magnesium. And this is a case when water is directly involved. And we'll stop it and try to come back. We try to continue as usually. Okay. So I will show you only one movie later. So there are other interesting cases of water. This is Camp Eliminase, which is kind of one of the key systems where people try to do protein design and try to create catalysis. And one interesting case there that we found is that you have a mutant which supposedly give you significant catalysis. And when we try to see why, it appears that it happened because of very trivial reasons which have very little to do with enzyme catalysis. In this system, you have proton donor, proton acceptor, glutamic acid, and if you try to make it unstable, it picks the proton better. This belongs to technique of ground state destabilization, which is not used by proteins, but I don't have time to tell you why. So now we go to the more entertaining part, which is proton transfer. Now, water could be a medium for a transfer of protons. A lot of papers were written about whether it's Eigen or Moses or other mechanism. I was never too excited about it, especially because people push at most what is called, okay, first of all, to show you systems in bacterial reaction center, there are pathways for proton transfer. In many other proteins, it's play a crucial role. And in many cases, there are water molecules inside. And early on, people suggested that the proton is transferred but by what is called the Grotus mechanism. Now, Count Grotus lived 200 or more years ago. He never knew too much on water, but he proposed really very insightful idea on the existence of water. And it was attributed to him something which is extremely easy to see, and it probably works in water, that when a proton transfer, it goes like this. And now, when it goes in a channel, the idea was that it goes in this way, like ducks turning their head, and you have to have water orientation as a control, the key control of proton transport. There are several major problems with this concept, which was accepted by almost the entire community because, first of all, it's nice to say Grotius mechanism, especially when you never study any real proton transfer in water or any other media. One problem is that if you have a very, very long channel, it will take huge energy to rotate all the water. So this is the Grotius mechanism, popular and believed by most people. Since we calculated proton transport very early in the mid-70s, I argue that it's mainly what counts with proton transport is the energy of protons at different sites along the chain, and clearly not the water orientation, which cost very little. So to demonstrate this, and most importantly, to really be able to model proton transport, we use our empirical valence bond model, which describe state like proton on one water as one force field, proton on the other water as another, and then you mix them to get the quantum mechanical mixing. Now, this model is extremely effective for chemical reaction with one proton transport. When we want to do it in a very long system, we design time-dependent all-atom model where it treats explicitly by this empirical valence bond the proton transport chain, and the rest of the system was treated classically and included in the PT Hamiltonian. This system was run by Langevin dynamics, so we could model up to even millisecond. And several key systems that we study. One of them is carbonic anhydrous, which has you heard about carbonic acid, which has this thing of transferring proton from the zinc to different acceptors, and this is measured experimentally. So you have a proton transfer chain. We model it in a very realistic way and could get the time dependent of being on different sites. And we reproduce the observed time dependent. A much more challenging case was cytochrome C oxidase, where even now we are not sure about the exact mechanism, but it's clearly controlled by proton transport pathways. And at a time, we try to see if the dynamics of their moving tell us something. So there are different pathways. And in this case, since it was very massive calculations, we replace the Langevin dynamics by time-dependent Monte Carlo. You could do it when you know the time steps, how long it takes to do a step, and we know the time step for proton transport from simulation when the barrier is close to zero, and then when it is higher, we use the Boltzmann probability. So we produce, I hope that this movie will do less problems. Yeah. So this is a proton transport in cytochrome C oxidase with this time-dependent Monte Carlo. And I think you could follow the proton. Okay. This is the same story. And here is the time dependent for different pathway, which we could simulate, explore, and ask questions. So there are different channels, D pathway, K pathway, and this is how you could model them. Now, to really shut down the Grotus community, we decided to take the gamma-sidon channel, which has extremely well-studied proton transfer mechanism and extremely well-transfer time for this in different pHs. So again, we use this empirical valence bond for the explicit water, electrostatic for the surrounding. And as you see, there is a very high barrier. So the barrier is for moving a charge across the channel. It is not for rotating the water molecule. So we simulated it. What you see here, one axis is the time. The other axis is the probability of being on different water molecule. I have a very nice movie, but it's in the wrong format, so I cannot show you. What is this? I don't know. OK, so now the last system for this controversy, when aquaporin was discovered, eventually they gave the Nobel Prize for it. It's a channel for water, not for protons. And in some stage, people start to ask why it blocks proton transport. And instantly, the Grotus community took over. And none of this community ever calculated electrostatic in protein or proton transport in water. But what they say is that there is a central motif called NPA of 2-glutamine, which was stated that this glutamine do not let the water orient. While it was quite clear that the glutamine control the energy of proton when it is in the middle. So after a few years, we decided to enter to the story. We model it, and we show that the electrostatic barrier is far, far bigger than any water orientation effect. We did it with different models. We reproduced the effect of mutations. And slowly, slowly, the Grotus people still say that it is Grotus, but they turn it to electrostatic. So now I will go to a model of water which even simpler than what we had up to now. How we are doing in terms of time? OK, we are good. So when we try to describe really, really large systems, we decide to simplify the protein. And the first simplification in 1975 was to replace the side chains by sphere. This was a model for folding with Michael Levitt. But then we decided, 15 years later, that the model has to have more electrostatic. And we have this coarse-grained model with simplified side chain, but also with terms that reflect the penetration of water, the surrounding water, and so on. The model was trained or calibrated on folding energy of many proteins. And it was used for for many systems, but one of them was the F0 part of ATPase, where the penetration of protons lead to rotation of the upper stack, which does the chemistry. So this has a main part with a ring of aspartic acid called the C ring. And attached to it, there is an A ring with arginine. And together, with the help of proton transport, this leads to a very nice motor. In recent years, the full structure of the A was also given, and we repeat our work. But the main idea was that we decided to get the mechanochemical work by this water proton transport model, which is, again, mainly energetics. It's the energy of the proton in different sites and not the orientation or any fancy things. And by calculating the energetics, and we succeed to build a landscape for the motion of the proton and the rotation and the pH dependent of the pumping. So it's a nice story that I will skip. We also did the time dependent. And last comment, there are systems which are even more interesting. There are systems where you have coupling of proton transport and external potential, like this edge V1. It's a voltage-activated proton transport. And we could also model this system when you change the polarization. First, when you have the system in the resting place, the proton cannot pass. I think it did not pass. But when you change the polarization, a barrier, which is due to arginine, moves and allows the proton to go through. Did it happen? Yes. So this is a nice, interesting story, which I would not discuss. Of course, a lot of people will participate in this work, and I thank all of them. And my conclusion overall that water are very important in biology, but it's not always the fine details which are important. And people like to talk on protein as being slave to the water. I don't know if you heard it. It was popular 5 years ago. Mainly Fraunfelder pushed this idea. It's completely incorrect. The water are part of the system. Nobody is slave of anybody. Thank you.
Thank you so much.